This method combines biochemical and behavioral approaches to help answer key questions in the neuroscience field about the impact of an acute kynurenine challenge in rats. The main advantage of the technique is that it provides a reliable and consistent method for the measurement of kynurenine pathway metabolites in rat brain and plasma. Demonstrating the procedure will be Carly Fabian, a student research assistant from my laboratory.
At the appropriate experimental endpoint after intraperitoneal kynurenine administration, thaw frozen harvested plasma on wet ice, and dilute the thawed samples with ultra-pure water. Transfer 100 microliters of diluted sample into individual containers of 25 microliters of 6% perchloric acid, and vortex and centrifuge the samples. Then, transfer 100 microliters of supernatant from each tube into individual 250 microliter microcentrifuge tubes for kynurenine and kynurenic acid determination. For kynurenic acid measurement from brain samples, thaw the frozen harvested brain samples on dry ice and individually weigh each sample on a precise analytical balance. When all of the samples have been weighed, move the tubes onto wet ice, and dilute each tissue sample at a 1:10 weight by volume ratio with ultra-pure water, followed by homogenization with a sonicator. Aliquot 100 microliters of each sample slurry to a new tube, then add 25 microliters of 25% perchloric acid for vortexing and centrifugation. Then,